So it's really my great pleasure to introduce Jen Adair today. First of all, I'll start with her her titles. So the FUNAS, she's fun as hell, but that's not what it means. So she's a fellow of the Ugandan National Academy of Sciences. So that's what FUNAS is. So Jen did her PhD at WSU, had very glowing reviews there. She moved on and did I think it would be called a postdoc at the National Environmental Sciences program in North Carolina. And then we were fortunate that she moved here to Seattle as a, I don't know if you'd call it a postdoc or research, sort of like a second postdoc, but called a research associate at Fred Hutch in Hans Peter Kiem's lab. And there she started really into the field of gene therapy. And she's really done some really fundamentally important work, both in lentiviral and gamma retroviral delivery to hematopoietic stem cells. She ran one of the first gene therapy trials out of the Hutch for glioblastoma, has been involved in many other cell manufacturing campaigns related to different gene therapy products with mostly stem cells and I guess some CAR T cells there as well. She then moved into the gene editing space and has pioneered some work in nanoparticle delivery of CRISPR reagents into hematopoietic stem cells and B cells, and even stepping on our toes a little bit apparently. Meanwhile, she's really been a leader in education and the field of gene therapy. She has many different roles within the ASG, the American Society of Gene Therapy. I think she was the founding member of what's called the Global Gene Therapy Initiative, which is really a lot about what she's going to talk about today. How do we get gene therapy, which is expensive to patients, both in the developed world and in the developing world. She's the board member of Caring Cross, that's their agenda, essentially. And she's also been involved in a ton of teaching and mentoring at all different levels throughout her career. So she's really a leader in many different arenas. The worst news is that she's leaving Seattle and moving to University of Massachusetts, where she's going to be the associate director of the Jorge Gene Therapy Center and the vice chair of a new department of genetics and cellular medicine. So she's going to keep on leading the field forward, a little bit further away from Seattle, but I'm really excited to hear Jen's talk today.
Thanks so much for that amazing introduction, David. You ate too much of my time, so now I have to talk really fast. Just quickly want to acknowledge that we do live, work, and play on the unceded ancestral lands of the Duwamish, Puyallup, Suquamish, Tulalip, and Muckleshoot nations. I really want to thank the original caretakers of this land that have kept it in such great condition and have provided the space for us to do the work that we do that I get to share with you today. I do have a number of disclosures. The two that are going to be important for this talk are honoraria I received from Miltenyi Biotech based in Germany. And I will highlight that work when I come to it. And as David mentioned, I am on the executive board of directors for a nonprofit called Caring Cross. It's an unpaid position, but we do a lot of collaborative work, and I'll highlight where that's important. This field of gene therapy is really booming. As of 2021, there were more than 500 gene therapy developers. We now have 34 gene therapies approved worldwide, and there are more than 2,000 in development right now from preclinical to pre-registration. And the FDA still expects to be approving double digits of these by the end of next calendar year. Now, the kind of gene therapy that you use really depends on the desired therapeutic outcome and the cell type that's required to achieve it. And the three flavors that are really prevalent in the United States, these are all the gene therapies approved here in the US, are those that are directly administered. This is typically with an adeno-associated virus. It can go into non-dividing cells if you know where those cells exist, and if they're in defined locations, or if they're in the liver, which is where AAV naturally goes. For T cells shown in blue and stem cells, these are going to be cells that we can manipulate outside the body, but their job in the body is to divide. So we really have to deliver genetic material into the DNA of these cells if we want it to be a durable gene therapy approach. And this is what area of gene therapy I have worked in. Now, as David mentioned, I came to the Hutch in '08. I was not a virologist. I was not a gene therapist. I am kind of a child of the Human Genome Project. I was in high school when they started it. I was in the second year of my PhD when they finished it. And leveraging that information for human health has been something that I've been very interested in throughout my career. But when I came to the Hutch to work in Hans Peter's lab, he said, we have this phase 1 clinical trial we've been funded to do for blood stem cell gene therapy in stage 4 brain tumors, glioblastoma multiforme in adults. And what we need to do is get this gene into hematopoietic stem cells, which are shown at the top of the hierarchy on the right. I'm going to call these CD34 positive HSPCs throughout the talk, just so there's no confusion. CD34 is a marker we use to purify these cells, but it is not unique to the stem cells at the top of the hierarchy. It really defines all of the cells in that purple box that I'm showing there on the right. And because these cells need to divide and we need to get these genes into their DNA permanently so that they're carried forward, the field had really turned to retroviruses or RNA viruses to do this. These viruses have an RNA genome. They attach to specific receptors on the cell's surface, administer their DNA cargo, which gets reverse transcribed and then integrated permanently into the DNA of cells, and then leverages our cellular machinery to make new virus particles. And this is what happens in nature. But unfortunately, CD34 positive HSPCs have evolved alongside viral pathogens, specifically to be resistant to viral pathogen uptake and transduction. So just a couple of the features of these cells that are going to be important for the work we talk about today. They're very rare. They represent about 0.8% of the total nucleated cells in our bone marrow, and they're very diffuse throughout the bone marrow. So really internal compartment, very spread out. They have a very tightly controlled rate of protein translation. These cells have a lot of transcripts sitting around waiting to respond to signals in the environment. They're super sensitive to amino acid level changes. As soon as they get a signal to start making some proteins and those amino acid levels drop, these cells go, oh, wait, I think I have a purpose. I'm going to become a T cell, and they sort of stop being a stem cell and differentiate. If they get infected with a virus that sort of commandeers that process and uses a lot of amino acids all at one time to make viral proteins, the cell goes, this is out of control, and it initiates a proteotoxic stress response and dies. These cells, because they haven't decided what they want to be when they grow up, also have very open chromatin. So when you make genetic changes in these cells, you have access to a lot more of the DNA than you do in a more differentiated cell type, which means there are long-term risks for genotoxicity. And these cells display both intrinsic and extrinsic pathogen responses. So they, again, are going to be very resistant to this viral transduction approach, as many, many people have validated in the field. And to get around that, these viruses have been pretty heavily engineered to get them to go into blood stem cells, either in a culture dish or possibly inside the body. And really, this is engineering the proteins on the cell surface of these viral particles, the glycoproteins in particular, to recognize different receptors that can be on stem cells, and then also essentially replacing all of the viral genes with therapeutic genes and preserving the capacity for these particles to carry reverse transcriptase and integrases that allow them to finish inserting this material into the DNA of cells. Very recently, as has been done by Bruce Torbett here and some other folks, these viruses can be engineered to actually recognize specific receptors on HSPCs to allow us the capacity to administer these viruses without having to necessarily purify the cells first. But administration of viruses, whether it's ex vivo or in vivo, elicits immune responses, again, intrinsically in these cells even when they're purified, and definitely extrinsically when they're administered inside the body. So manufacturing of these cell products is complicated. This is actually what is done for all of the stem cell products that you saw on the previous slide that have been authorized in the US. Involves either getting these cells directly out of the marrow or mobilizing them into circulation, purifying them, culture to stimulate these cells to divide so that they'll more readily take up the viruses and do these processes of integration and reverse transcription. And then because you've done a pretty heavy manipulation of the cells outside of the body, you have to give them a competitive advantage when they go back in. And the way we do that is by treating the patient with chemotherapy to knock down the endogenous stem cells in the marrow and create space for these engineered cells to have a chance. And all of this is done in what we call an International Standards Organization, or ISO Class 5 clean room, which we're fortunate to have both here at Children's and at the Hutch. So this was that first trial David mentioned for glioblastoma. My job was to take a gamma retrovirus that carried a gene called MGMT P140K and put it into blood stem cells. And what this would do is give the patient with a brain tumor a blood system that was very much shielded against a really intensive chemotherapeutic regimen that could knock down the growth of the brain tumor. This was successful in terms of a gene therapy approach. We were able to see the gene expressed. We could see the patients able to receive more doses of this chemotherapy and at higher levels than were previously done. And we saw an overall response in their brain tumors. Life was good. This has moved forward, and it's still in clinical development in a commercial organization based in Australia. But at the same time, lots of groups were showing that because we can't control where these viruses insert their genomic information, there is a risk of genotoxicity, in particular with the gamma retroviruses. These like to insert near gene promoters, and if you have a strong viral transcription rate and you're near an oncogene, you can turn on that oncogene. And this is what happened in X-linked severe combined immunodeficiency in a clinical trial in France that resulted in cancer in about 20% of the patients who had received this blood stem cell gene therapy. So the regulatory body said, you have to mark. You have to be able to trace these cells. You have to look at where these viruses have integrated in the patients that you're treating on these trials. And this led us to develop an assay called retroviral integration site monitoring. For those of you I've collaborated with on these projects, I apologize, this is the only slide you're going to see on this. It's just to say I've had the privilege of working with a number of bioinformaticians. We not only were able to do this for safety monitoring in the patients, but then we could leverage that information to learn about what cells are dividing, how many are there, what sorts of other cells they're becoming, and it taught us a lot about what hematopoiesis looks like in patients after gene therapy and after transplant. This led to at least 8 publications, which are shown at the bottom, and the PhD or successful papers of all 3 of those bioinformaticians that you see. Lauren Schefter, I think I just shared with someone earlier, it's actually someone I met at the Starbucks drive-thru at the AGC building. She just said, where are you headed, work? And I said, yeah. And she said, where? And I said, Fred Hutch. And she said, what does your lab do? I said, gene therapy. She said, I'm really interested in bioinformatics, but I haven't been able to find a job. So I gave her my card. She volunteered for about a month, and I hired her right away, and she produced the graph that you see on the right. This led to what I call the heyday of my career, blind ambition and joy. I was living the life, as David mentioned, advancing gene therapy translation for blood stem cells across a number of different disease conditions, lots and lots of papers. This looks like a beautiful scientific trajectory, and it feels awesome when it's happening. And then someone asked me to translate a trial for patients living with HIV. And this was really a turning point for me for a number of reasons. The first was that at this point, we had sort of transitioned from gamma retroviruses to lentiviruses because of that genotoxicity issue. The lentivirus we're now using is an HIV-1 derived lentivirus. So the first thing I thought, scientifically speaking, was how the heck am I going to get an HIV-derived lentivirus into cells that are treated with antiretroviral therapy for HIV? And what the heck is the safety concern if there's already HIV in these genomes and potential recombination with the engineered HIV that we're going to put in? Am I going to be the person who is the epicenter of like a new supervirus epidemic that I don't want to be part of? So I started to read in the literature, and then someone said, oh, this CROI meeting's happening in Seattle this year. You should go. It's all about opportunistic infections and retroviruses. You could totally learn a lot. And I thought, awesome. And day 1 of the conference, first speaker gets up and puts up this map. And I had a totally existential crisis in the back row of the conference because everything I had done before was for rare diseases or ultra-rare diseases, hundreds of thousands, maybe hundreds of patients. This is 40 million people. And I am very ingrained in the gene therapy world. I've published all these papers. I've gotten to give a lot of talks. I knew that these are the places that were actually developing gene therapies, that were moving into clinical trials, and there was a complete disparate delivery model here. So not only do we have to scale massively to actually make a dent in this global health burden, but we're doing it in totally the wrong places to learn the things that we need to learn in order for it to be successful for the people that it's going to matter for the most. I then dug myself a deeper hole. I went back and I looked at how many clinical trials for some type of gene therapy to treat HIV had been registered since the mid-90s, and the answer was more than 36. And the number of those in 2014, and the same answer is the number of those today that have actually taken place where the majority of patients live and where the epidemic started is 0. So I decided to take a minute and take a step back and sort of think about how I could gatekeep differently in my career. And this meant a couple of years with no publications, kind of trying to figure out how I was going to satisfy this very dissatisfying number with what I am capable of doing. I will also say, this is a very recent review that Kevin and I just published in Science Translational Medicine this year in May as part of a special series. I'm really proud to say this series includes more authors from underrepresented low and middle income countries who are interested in gene therapy than in the history of the journal of the entire Science Family Journal series. And this review is one of those key papers. We highlight 6 countries, case studies, where they're actively pursuing gene therapy research and development, for which we have authorized gene therapies in high-income countries, but for which those countries actually have the highest burden of patient disease. And we can learn a lot from these spaces. Now, back in 2014, when I was having this existential crisis with HIV, the first thing I thought of is, I might be able to help. Really, this clean room process, the clean rooms themselves cost multi-millions of dollars, and this complexity, the number of personnel you need to purify the cells, to culture the cells, to transduce the cells, to do the quality control in the cells and release test the cells is a huge burden that's going to be difficult to overcome. Can we automate this? And at that point in time, there was a possibility in an off-the-shelf device, the Clinimax Prodigy, that was developed by Miltenyi Biotech. On the left is the Clinimax Plus. This was the instrument that we still use around the world a lot today to purify CD34 positive cells from bone marrow or leukapheresis products. But you have to do some platelet washing, you have to incubate the cells with the beads that detect CD34, et cetera. And so Miltenyi had developed the Prodigy to automate those upfront steps. So there was a centrifuge capacity, there was the ability to regulate some temperature in this device, because you got to incubate the beads at 4 degrees Celsius. And there was some sensor detection that allowed you to monitor phases of the blood cell pellets in this device. So we approached Miltenyi Biotech and I said, can I buy a device and are you okay with me engineering it and maybe reprogramming a little bit? I'd like to see if we can automate this manufacturing process for lentiviral-based blood stem cell gene therapy. And they agreed. So with this pretty awesome team of 2 research techs, Davika Chandrasekaran and Gabriela Skia-Hughes, a postdoc, Dr. Sarah Kubak, and a really amazing animal technician who specializes in non-human primates, Veronica Nelson. We bought the Prodigy. This is one of our first bone marrows that's running on this device. And it took us 2.5 years, but we did successfully automate this process. And we were able to show that these products could in fact meet US FDA standards for an academic research center to infuse them as part of a phase 1 clinical trial. Here I'm just showing 2 different apheresis products, meeting all of the release criteria that our products at that point in time had to meet in order to be infused. And then really critically, we actually transplanted 2 monkeys with cells that we manufactured as if they were gene therapy patients on this device, except we totally myeloablated those monkeys with total body irradiation. So if there was any contamination or problem with this cell product, the monkey actually wouldn't have survived the process. Not only did they survive, but they engrafted just as well as monkeys who had received products manufactured in the standard process. And this was really great, and I started to feel like maybe I'm coming back into my heyday. I'm starting to climb out of this pit that I had felt of existential crisis. And then I did the dumbest thing in the world. I went through my white savior era, where I was like, I have your solution. It's this automated device. And I went to Brazil and India and Uganda, and I talked about this device. Quick side note, has anybody seen this map before? It's called the Peters projection. This is a map that accurately represents the differences in size between our continents in reality versus the globe, which distorts to make the US look bigger than it actually is relative to places like Africa and South America. And it's fun because you can find this map on a website called The Conversation, 5 maps that will change how you see the world. And conversation is really what happened when I went to these spaces that made me realize I was in my white savior era and there was a lot more work to do, and maybe I am not the person with all of the answers. Because what they showed me was that there's a heck of a lot more problems with this complicated manufacturing process than what we had talked about or what I thought we needed. And the reality is that there are some spaces that are going to be able to adopt certain changes more quickly, and other spaces where we have to innovate in completely new ways if we're going to make progress. And there are ways that we can do that together. The one thing I would impress, especially on the trainees in the room, is that talking to people solved some of the greatest problems in this space and really ignited the next phase of my research career. Now, the field knows that the ultimate destination here is a shot in the arm, something that's akin to vaccination. So we need some syringe that's going to have some gene therapy tool in it that we can literally just inject into a vein without any other treatment required, that will somehow find the blood stem cells, administer the correct genetic engineering, and then let those cells take off and do what they're supposed to do without having to chemo condition, without having to screen a patient, without having to worry about immunogenicity. And that's a laudable goal, and we should be working towards that. But what I also learned in my conversations was that there's a lot of unexplored runway between where we are now and there that can make a lot of transformative advances for folks in different spaces. And this is really what I'd like to spend the majority of today talking about. Most of what you're going to see is unpublished research. There's a few publications. And really, I'm going to take you through sort of what I've simplified to a 3-step process. There's a whole lot more runway in between these 3 spaces. I'll talk to you about the automation of this process and where that can be useful. I will also talk to you about developing simpler ex vivo processes, simplifying lentiviral gene therapy to make it just more affordable and accessible in research spaces and clinical translation spaces. How we're advancing technology, as David mentioned, with nanoparticles to simplify CRISPR delivery, and how both that simplified lentiviral vector gene therapy and simplified CRISPR delivery are setting us up for that shot in the arm, which is probably going to happen in the next 20 years or so. But we will also talk in the 4th vignette about the ways that we have to think about actually successfully accomplishing that in patients, and some new models we've developed to address some of those issues. So vignette number 1, where has that automation on the Prodigy gone since we published it in 2016? And the spoiler alert is India and South Africa are the farthest reaches. South Africa, David Britton in particular at Albert Alberts just reported the first transplant of patients with CD19 CAR T cells that were manufactured on this device in the last few months. We've made a lot more progress in India. That first clinical trial result is in review right now. It's called Velcart, and I'll talk a little bit more about it. So upper left is that first paper that we published on stem cells. Miltenyi Biotech knew that it would make the most money in immunotherapy. So they were taking our programs as we were developing them for stem cells and translating them into T-cell programs that are now part of, if you're familiar with this instrument, what is called the TCT suite, the T-cell transduction suite on this device. And you can see that there are 3 T-cell papers to our 1 stem cell paper that prove just how useful that was going to be in the commercial sector. Now, Boro Dropulic, who is a co-founder of the nonprofit Caring Cross, but was originally founder of a company called Lentigen, which manufactured lentiviral vectors, that got acquired by Miltenyi Biotech so that they could build out essentially gene therapy, all of the gene therapy product suite that anyone could need for manufacturing out of their company as a single supplier, was actually the first to show that if you had 2 prodigies in a totally gene therapy naive clinic, one was in Ohio and the other was in Russia at the time, and if you used the same protocol, the same process, and you had the same reagent source and stream and material source and stream, that you could manufacture products that were equivalent and that had similar behavior in patients, both of which were efficacious. And this was really the first time people started to realize the value of automation in extending international access to clinical protocols. But of course, the majority of that is being done in high-income countries that can pay a lot of money for these devices, reagents, materials, and supplies, and where the commercial companies that can buy this device can justify it because they're going to charge hundreds of thousands of dollars for these T-cell products to private insurers. What we did was go in the other direction. We said, rather than go to a high-income country where you have health insurance, let's go someplace where the majority of the population's in a self-pay healthcare system. And let's see how affordable do we have to make this gene therapy to make it work. So we've been collaborating with Vikram Matthews at Christian Medical College in Vellore, India for a number of years now. Vikram has an amazing team. They got a prodigy. They started manufacturing CD19 CAR T cells, built the qualification for that. Now, India is a unique case because they have a high spending capacity but a low workforce cost. They have a regulatory infrastructure for clinical trial development and academic research already in place. So all we had to do was get them to approve a gene therapy trial, which was no small feat. And I say we, like I had a lot more to do with that than Vikram. The reality is Vikram and his team were incredible. The biggest lesson we learned was that in India, there's kind of a sequential process for approval, and the hardest part was them deciding who was gonna do the final sign-off. It turned out that every regulatory representative was like, I don't wanna put my name on the line because if something goes wrong, then the government's gonna come after me. So we came up with a new model where they could essentially have co-signers so that people would share equal responsibility and they felt more comfortable with that. But this got the trial through. As I mentioned, Vikram has treated 12 patients with great results. They're gonna publish that study. But importantly, we were able, because this is a self-pay healthcare system, to talk about exactly how much it costs to make these products. $35,107 is still a lot of money per product. It's 1/10 of the cost of what we pay here in the United States for commercial CD19 CAR T cell therapy. And as you can see by this pie chart, the majority of that is reagents and consumables that have to be imported from high-income countries. So India is now moving towards local manufacture of these reagents and materials to reduce the cost further. We're very confident that we can get under $15,000 a product for these. But also, this excludes the cost of the lentiviral vector that conveys the CAR. And most of you who might be in this space know that it roughly costs about $2 million just to get out the door to get enough GMP-grade vector to move forward. And that's because all this engineering we did to those lentiviral vectors, we still can't make them in a test tube. We still need a cell to put them together for us, and it's very inefficient. And so we have to make large quantities and purify it away from the rest of the cellular material to have a virus that is qualified to be used for gene therapy trials. And this means that for stem cell gene therapies, a single batch of lentiviral vector can maybe get you 20 patients, maybe. David's raising his eyebrows, it's usually 10. In the CAR T cell space, it can be a little bit more closer to that 20 mark, but it's still in a tremendous expense. So this brings me to vignette number 2, a project we've been working on to simplify lentiviral vector gene therapy in HSPCs. And really, we're targeting how can we make these viruses more potent so that we need less virus. If you have one batch, can we use less per patient? Can we shorten the time it takes to manufacture and use fewer reagents and materials to essentially drive down that cost a little more? This is the work that we're doing in partnership with Caring Cross. The financial contribution of Caring Cross is that all of the vector you're going to see in the following studies was donated in kind by Caring Cross. And this project has been led by 2 Ugandan postdocs, Dr. Lois Baiga, who came to my lab between 2021 and 2023. She is officially the first gene therapy researcher in Uganda. She's now based at the Joint Clinical Research Center in Kampala there, and this is a picture of her in my lab. And then Rachel Bachaite Chiune is actually an SCRI Invent Fellow who is in my lab carrying this project forward. So we talked about these engineered lentiviruses and their life cycle, and really the first step of that is this virus recognizing a receptor on the cell that you want it to go into. This picture kind of shows the viral particle fusing with the cell membrane, but in reality, these vectors actually go into an endosome and then fuse with the endosomal membrane in the cell. And this process takes anywhere between 2 and 4 days. It leans closer to 4 than 2. The cells are usually purified and or thawed out of cryopreservation and put into a stimulation culture because they don't actually express the receptors that most of our engineered viruses use to get into these cells. I'll talk about that in just a slide. Once they express receptors, usually within about 24 hours of this culture, we add the virus twice to try and give us enough of a chance of getting into these cells. Generally, the number of virus particles per cell is somewhere between 20 and 200 per hit. And then you wait, because now you need to let the virus get in, you need to let reverse transcription and integration happen before you can harvest these cells and prepare them for infusion. Now I'm showing at the bottom, vesicular stomatitis virus glycoprotein is the most commonly used glycoprotein on these engineered HIV lentiviral vectors that are used in clinical trials. Every one of the gene therapies authorized in the US that uses a lentiviral vector is using a VSVG pseudotyped lentiviral vector. And this glycoprotein recognizes something called the lipoprotein or low density lipoprotein receptor, LDLR. Now, LDLR is expressed on pretty much every cell in the body, not stem cells until you culture them for 24 hours. But this is a reason why we can't just use a VSVG lentivirus inside the body, because plenty of other cells would take up that virus before it ever saw enough stem cells. And the stem cells circulating in our body aren't necessarily expressing LDLR. While I was in the Cheme lab, we developed a second glycoprotein called COCL. This is similar to VSVG, it uses LDLR. One of the advantages of COCL is that it's not toxic to producing cells. So the issue with VSVG is you have to retransduce or retransfect the producing cells every time, because expression of VSVG and the other particle components is toxic. So the cells will make the virus for a little while and then they all die. But COCL is not. So we can actually create a working cell bank of producer cells that you just thaw, they'll make virus for you, or you just put in your therapeutic transgene, your viral genome, and then it will make virus for you for a while, and then you can get rid of those cells and you can do the same process over again. So it makes manufacturing a little bit easier. But it also uses that LDLR receptor. There's also baboon envelope viruses, which use the ASCT1 and 2 receptors, and then there are measles virus or paramyxovirus family members, which can use a variety of receptors, including CD46, SLAM, and another receptor called Nectin-4. So Lois, when she joined my lab, said, I want to first compare VSVG and COCL to see how low I can go in the amount of virus per cell in a given transduction protocol, and which one will give us better transduction. So we just used a virus that encoded a green fluorescent protein, and she ran an experiment where she tried different viral levels, and she found that COCL was actually far superior to the VSVG that we got from Caring Cross. Now, these VSVG results are lower than what most people would see with a GFP vector, but the COCL results were actually really good, and the COCL virus you can manufacture at much higher titers. So we felt like we were just going to go with that. So the next thing she tried to do is see if she could take this low viral particle number of 20 viral particles per cell and increase the transduction efficiency in HSPCs. And so she tried a couple of different conditions. Mock just means no virus was added. Culture is just the virus added to the cells in culture, as I showed before. Spinoculation is slow centrifugation of the cells after you add the virus to try and bring the virus in close contact with the cells in the bottom of the centrifuge tube. And then poloxamers are what most people in the lentiviral space know as an enhancer of lentiviral uptake. I'm showing on the right, these are just tri-block copolymers. They have a hydrophobic core surrounded by 2 hydrophilic units. They're essentially surfactants. They just change the electrostatic positive and negative charges on the cell membrane in the virus to allow them to come in close contact and just hang out with each other for a while. What this means is that if the cell is sampling its environment, it will take up those virus particles just by micropinocytosis and not necessarily require a receptor for entry of the virus into the cell. And what Lois found was that the poloxamers greatly enhanced the COCL, and that was great. I'm not showing the data here, but we didn't see as much with the VSVG. And what she thought was interesting was that COCL and VSVG are supposed to use the same receptor. So either they're using a different receptor or there's some feature, physiochemical feature of the COCL envelope that actually is facilitating post-entry transduction more so than VSVG, even though they're both using LDLR. So she decided to look at the receptor expression, and then this was work that was actually completed by Rachel Cheune. We monitored across 10 different donors the expression of these receptors, CD46, LDLR, Nectin-4, and SLAM, from the cells immediately after their thaw all the way through the first 24 hours of our stimulation culture. And they saw that LDLR, the blue line, does come up over those 24 hours. That 24-hour mark is kind of where it flatlines for the rest of the culture. But CD46 is really expressed constitutively at the top. And so she went back and she said, well, if COCL is sort of enhancing the viral's post-entry transduction, And the cells are already expressing CD46, which is what the measles viruses or the paramyxo family of viruses uses. Could we combine those two into the same envelope and actually create a hybrid that would take the best of both worlds? And we thought this was possible. People have done this with measles viruses. I know Bruce's lab is looking at NIPA to actually engineer viruses to detect specific cell types. So we thought that this could be pretty pliable. And I'm not allowed to tell you all of the things that were done. I can tell you that if you ask Caring Cross, they can provide you with some of these vectors to test. We essentially produced 4 hybrids, F1, F4, F9, and 11. They are all various mixes of the paramyxovirus coccal envelope glycoproteins. And what we saw when we compared to standard coccal across 4 donors is that we could get very good transduction at as low as 10 viral particles per cell. And we could do this adding the virus immediately after we thawed the cells. There are no poloxamers. This is just media and 3 growth factors, which is the standard minimal culture condition. Now, we know that they can use CD46 at this time point, but they don't have any LDLR. So Rachel had a question, what if we waited just 24 hours till we had peak expression of both receptors, what would happen? And the answer is it's still not as good as adding the virus right after we've thawed the cells in culture. The best results we got were when we put the virus in right after we got the cells, just let it sit overnight and harvested the next morning. Showing VCNs here, we have GFP expression data, we have colony-forming assay data. There's no hit on viability at all at these viral loads. And the funnest result we got was that we get pretty predictable transduction. If you use an MOI of 10, 10% of the cells get transduced. If you use an MOI of 20, 20% of the cells get transduced. If you use an MOI of 50, 50% of the cells get transduced. And that's very reliable from donor to donor. So they both have taken this process of up to 4 days and condensed it down to 1 with limiting viral particles. You don't need any additives. It's incredibly short. It's very efficient. We now have the same hybrid vector manufactured with a hemoglobin gene, a beta globin gene for the treatment of hemoglobinopathies. We have made GMP-grade hybrid vector, and the mouse studies are currently cooking. This has been picked up by the Washington Research Foundation, who's funding Rachel's continued work in this project. And we continue to partner with Caring Cross, who has a whole other set of research on these same hybrids in T cells for the production of CAR T cells that they have also managed to reduce the manufacturing down to a 24-hour period for. So look forward to sharing more of that data with you. So that's where we are with lentiviral vectors. Let's talk a little bit about CRISPR. This is work that has been 5 years, a ton of effort. It was started by a postdoc in my lab, Dr. Raisa Shabazi, who's now faculty at the University of Indiana. It's been carried forward by 2 postdocs, Dr. Daniel Lane and Dr. Halima Sidiya Malik, who is also an SCRI Invent Fellow here, and 2 graduate students, Rachel Cunningham and Karthikeya Gutamakala, who are in the M3D program at UW. We did spin this out through funding with OrbiMed Health Partners into a company called Areta Inc. that provides sponsored research funding for this work. So when we talk about CRISPR approaches in stem cells, the manufacturing process is just as complicated, if not more, as what you saw for lentiviral vectors, where we can just add a lentiviral vector into the dish and it will kind of do what it's evolved to do and get into the cells. CRISPR complexes are bacterial derived and they have no mechanism of cellular entry and delivery into the nucleus. So the way that that's typically done is with electroporation. Electric shock of the cells opens up the cell and nuclear membrane, and if you have a high enough concentration of these complexes, they will diffuse into the cell, and then you can put nuclear localization signals on the protein itself to get them into the nucleus. That's just if you want to make insertion or deletion mutations. If you would like to insert new genetic information, you either have to use an engineered version of the CRISPR and provide that template, or you have to add a viral vector, typically an AAV, that carries the genetic information with flanking regions of homology to the DNA where you want it to insert, which means we haven't gotten away from using viral vectors in this process, and we've now added additional steps and additional reagents. And this is the process that's being used for the authorized CRISPR gene therapy, Casgevy. It is just the electroporation of CRISPR molecules. You don't need an AAV in this case, but there are multiple other approaches for sickle cell disease and other applications where they are using both electroporation and AAVs. And all of it is still done in an international standards organization, Class 5 clean room. So I told you at the beginning that these CD34 positive HSPCs are very resistant to viral infection, pathogenic infection, and genetic engineering, and that includes from CRISPRs. They are very sensitive to amino acid levels. So when people tried to electroporate in messenger RNA, if they were not culturing the cells ex vivo, they got very little transduction efficiency or CRISPR gene editing efficiency. The cells ex vivo will make the mRNA into protein, but they almost immediately start differentiating because they've started to change their amino acid levels. So it was learned early on that more than likely we were going to have to deliver CRISPR as its ribonucleoprotein complex, which is a piece of RNA that tells the complex where to go in the DNA, and then the protein piece, which can bind, unwind, and cut DNA, which is actually going to introduce those genetic changes. And this is complex cargo. There's a lot of nanoparticles, our mRNA vaccines for COVID, for example, which are positively charged, very good at carrying in negatively charged nucleotides. There isn't a lot that can carry both an oligonucleotide and a protein at the same time. So we had to kind of think about how we could build this from scratch. And this was the work that Reza really pioneered. We published this in Nature Materials in 2019. Our thought was, we need a particle that can carry this complex cargo, and in an ideal world, even though Cas9 is mostly what people use, there were lots of other CRISPR systems. We were just starting to see the base editors and prime editors, engineered version, fusion versions of these proteins coming out. So you'd want a system that you could just, in theory, modulate any CRISPR system onto with pretty reliable efficiency. And so what Reza came up with was a gold nanocore, about 20 nanometers in size. We can just end modify with a thiol, a terminal thiol, the guide RNA. And this is done pretty regularly to preserve the stability of these RNAs inside cells. And that will allow a covalent interaction between the thiol and the gold ions on the surface of this core. Once you have that guide RNA anchored, you can add the nuclease protein. It will form its natural complex just on the surface of the particle. So you don't have to engineer the CRISPR to get it to load onto this particle. It should just happen naturally. And then you have a particle that can deliver RNP. We can then load polymers, specifically positively charged polymers like PEI and others. This will allow us to not only facilitate cellular entry and exit through endosomes, but it gives us a really nice positively charged bed to layer things like DNA templates for things like inserting new genes into these cells. And so we called this a first-generation CRISPR RNP. Reza showed that we can make these particles. They are red in color. The first use of gold nanoparticles was actually to make red stained glass in European medieval Europe. So when you look at red stained glass, you're actually looking at gold nanoparticles. When these particles lose their size or stability, they change color to blue to black. So it's a very easy visual indicator of whether or not they're usable. They're stable at room temperature or refrigeration at minimum. And things that are really close to the surface of the gold are quenched in their fluorescence, whereas things like the DNA on these particles that are really far away can actually fluoresce even after it's been loaded. And this allows us to monitor where cargo is going, how fast it's getting in, and how fast it's being released from the gold cores in cells. And this was work that we included in that paper using confocal microscopy, where we basically showed the particles are being taken up by these HSPCs through micropinocytosis within 30 minutes of being added to a dish. And within 6 hours, we can see the RNA cargo in green and the DNA cargo in red unloaded off of these particles and dispersed throughout the cell. But importantly, Reza also showed that we got gene editing. Now, what was interesting was that we got better editing with a nuclease system called Cas12a. The big difference is it's smaller, the guide is a little bit different, and this nuclease makes a staggered-ended cut, a sticky-ended cut, versus Cas9, which sort of does a blunt-ended cut. And so we didn't really understand why Cas9 wasn't working as well, but we saw gene editing with both formulations. That was one of the questions that we had, but we also wanted to know, is it possible for us to administer these nanoparticles in vivo? And to do this, we turned to a humanized mouse model. Rachel in the lab actually showed that the majority of the CD34 cells in this mouse model are in the bone marrow, primarily the iliac crest. That's a pretty fragile bone. You wouldn't want to stick a needle into it. You'd probably break the mouse's pelvis. So the next best bet is into the femur through the kneecap. Or we can mobilize stem cells using the same pharmaceutical reagents that we would use in a clinic to move stem cells from the marrow into circulation. So the first thing we did was try to mobilize these mice. We injected the particles at 2 different doses. We could see gene-edited cells in the peripheral blood about 2 weeks after these nanoparticles were administered, and then never again, which means that these particles are definitely getting into some cells, but it's not the stem cells. They're not long-lived. And that's not unexpected. We haven't done anything to tell the particles specifically to go into a stem cell. But when Rachel injected the same particles intramarrow or intraosseously, we saw a nice stable gene editing in the peripheral blood out to 20 weeks, especially when we used, again, the Cas12a loaded particle, but not the Cas9. These are also non-toxic. The mice do much worse with a mobilization regimen than they do with a nanoparticle injection, even though it's going into the kneecap. We've now given hundreds of mice these particles, really, really high doses. They're cleared through the hepatobiliary system. They go through the liver and essentially are excreted in the feces and urine. There is some gold dose that gets retained, especially when we do intraosseous injection in the bones, but it's considered non-toxic, and the mice don't display any phenotype associated with those nanoparticle doses being there. In a lot of work that you can now read in a preprint on bioRxiv, Daniel Lane and Karthikeya Gautamukula did a lot of physiochemical analysis to figure out what the heck was going on with Cas9. And this is the culmination of that entire paper's worth of research in one cartoon. What we were expecting to happen on the left is not what was happening. Due to the physiochemistry of Cas9, we were getting only about 4 RNP loaded per particle instead of 50, which is what we had anticipated. And that had a lot to do with the guide structure and the stability of the complex under the conditions we used to formulate the particle. They went on to create a second generation version of this particle that recuperated that, so we're able to load 50 RNP per particle for Cas9. We could restore editing in cell lines, but when we put it in primary cells, now it gets stuck in endosomes, meaning that we've changed something about the physiochemistry of the particle that now is interacting differently with the cell biology in these target CD34 cells. In new work that Karthik is drafting right now, and we hope to have in preprint later this year, he has tuned the nanoparticle cargo, including the polymer-based cationic component, to achieve peak editing, to create a third generation particle that not only carries the number of Cas9s that we would like it to, but it can now escape endosomes in primary CD34 positive HSPCs, and we can actually get really nice gene editing levels in these cells that are equivalent to our RNP controls by electroporation. And where are we hoping to apply this work? This was my Rich James slide, and then he went out of town and didn't even show up for the talk. I hope that's in the recording and he reads it, he hears it later. We have used this to engineer hematopoietic stem cells in a non-human primate, primatized mouse model called Mr. G or Mr. G6. This is going on preprint as soon as bioRxiv validates it and approves it. This is work by another grad student in the lab, Jack Castelli. He designed a Cas12a that could do this, purified the NHP cells, showed that he could edit and introduce a broadly neutralizing antibody against HIV called 1074, and he can transplant these cells in mice. They do form B cells, and those B cells retain this edit and express the antibody when in the presence of the antigen. So very exciting work that we hope will create new opportunities for us to approach diseases of global health burden using this nanoparticle formulation. So in just the last few minutes before questions, we have a simplified lentiviral vector that we know can get in really quickly. We have a nanoparticle that can deliver CRISPR within 30 minutes if it can find the right cells. We're slowly inching our way towards this shot in the arm kind of thing, but there are still a lot of other problems in the space that we haven't answered. And one of those is, how the heck are we going to get access to enough hematopoietic stem and progenitor cells? The mobilization regimen that's most used clinically involves filgrastim plus plerixafor, 2 different agents. The mechanism of action of filgrastim is to make these cells divide till they fill up the marrow so much that they spill out into circulation. So you might be able to get a lot in circulation, but they have nowhere to go back to because the house is already full. So we have to think about ways to mobilize these cells that sort of vacate the niche, make them accessible for engineering, and then allow them to just go back and start performing hematopoiesis. But we don't really have great models for how to assess this or how to determine which mobilization strategies are going to work. So in the last year, a postdoc that I picked up from Justin Taylor's lab, he moved to the University of Virginia, Dr. Grady Gastelum. I had presented this dilemma to him, and he said, give me a couple months, and let me see if I can come up with something. And so I said, fine, there's a paucity of data. Really, the questions I want to know are how many of these stem cells can we mobilize relative to the available pool of total stem cells? What fraction is going to be available for engineering? And how long is it going to take after they go back for them to start making different blood cells? What does the kinetics of that look like? So we know when to check whether or not the gene therapy is working. And I want you to do this as if there's no chemotherapy conditioning and no selective advantage following the genetic engineering, which is the hardest problem that we have to solve. So he comes back and he says, well, the only thing people have done is called the parabiosis model, where you sew two mice together and you mobilize one and look at how many of the cells from that one ended up in the other one. It's an intensive surgical procedure. The method is not clinically relevant to what we would do in patients. This is a super high burden on the animal, and I basically said, no way. Let's think of something else. So what Grady said was, there's this other wild-type mouse model. It's a C57 black 6, and they make a syngeneic version of this, which has just a little amino acid difference in a molecule that's on all white blood cells called CD45. So there's a CD45.1 allele and a CD45.2, and there's an antibody designed to recognize the difference between those two CD45 molecules on the surface of all white blood cells in this mouse. So what Grady proposed was, let's mobilize both males and females from each of those groups. Let's just take as much blood out as we can and transfuse it into the other allele right away. That's sort of the best model we could come up for clinical relevance. We somehow convinced our IACUC to let us do this, to take hundreds of microliters out of these mice. We just have to give them some fluid reconstitution, and then we do it literally within a couple of minutes. And then you get to see the opposing allele cells, and we're infusing a whole bunch of blood. So there's a lot of cells initially that then go away, and we monitor over time, especially the short-lived cells, the cells that, like granulocytes and neutrophils, that are going to turn over really, really fast, because those will not last, but if they're coming from stem or progenitor cells, they will. And we can also monitor long-term cells over time. So Grady has since proven this model. Here we're showing in green the plurixiphore mobilized animals versus the vehicle-treated animals, and you see lots of these cells. Quick fun fact, mice don't have CD34, so the marker set we use is called LSK for lineage negative, SCA1 positive, c-KIT positive cells. But LSK equals HSPC. So we see a lot of them in circulation. We're not really seeing them come from the spleen or the liver, which are known reservoirs for hematopoiesis in mice put under duress. But we do see them coming out of the marrow, and in particular the tibia and the iliac crest. So we're getting some cells out from the marrow, definitely not all of them. It's not even statistically significantly different with this sample size relative to the vehicle-treated controls. But it was enough for us to get started. Grady then went on and actually did the transfusion experiment using a mobilization agent called plurixiphore just by itself. So you administer the plurixiphore at a single dose of 10 mg per kg. 2 hours later, you harvest this blood and you transfuse it into the opposing allele. And we watch, and then as a positive control, we actually took the same mice and did a full-on bone marrow transplant, total body irradiation, everything, to show what those kinetics look like. I'm just showing the first 6 weeks of data here. We've carried this out now more than 10 weeks. It's going to go to 16 weeks in total. The red is those positive controls. Obviously, in a transplant situation, you expect to see a ton of donor cells take off. Green is our plurixiphore mobilized cells, and blue are our vehicle-treated animals. So we definitely see a higher engraftment level with the plurixiphore treated, but it's low. It's less than 0.5% of the total cells. In terms of fraction. On the right-hand side, though, you can see the absolute number of these cells is staying right around 2,000, which is about how many we had mobilized in the animals when we did the initial plurixiphore injection and took the blood at peak mobilization. So we're seeing sort of a stable level of cells. The next question for us was, what are the different lineages that are represented by this strategy? And we see a little bit of everything. We see B cells and T cells and granulocytes and neutrophils and red blood cells coming out of this model. For the plurixiphore mobilized animals, it's always better than the vehicle-treated controls, but at these animal sample sizes, it's not always statistically significant. But what we think this tells us is that we definitely have a model that's going to be more clinically relevant to assessing which mobilization strategies are going to be the most effective. at essentially getting us towards this in vivo genomic engineering, or even potentially a simplified ex vivo process. So Grady's moved on from Plurx4 alone to include a number of other agents that have mechanisms of action where they actually either agonize or antagonize the molecules that make stem cells stay in their niche, as opposed to telling them to divide and fill up the house. So these should all promote exit of the cells from the marrow for a short period of time and then return. They have wildly different kinetics of activity. Grow beta is 15 minutes from treatment to peak mobilization. W106 is about 4 hours from administration. And so we are in the process of actually doing a transfusion study with all of these right now using this model. And an ideal regimen will mobilize the highest number with the greatest predictability, work in a time frame consistent with being able to treat patients in 1 day, and permit the return of these cells to the niche where they can contribute to hematopoiesis with kinetics that we can reliably measure, so we know when to check whether or not the gene therapy is working in this context. So I hope I've convinced you that there is quite a bit of runway. Anybody who's in the gene therapy space, I hope this has given you ideas of other things to study. Please feel free to take those ideas and run with them, because we need more people thinking about this problem of simplification and access and affordability in order to actually solve these problems. And for all of 2024, all of my lectures, all my publications are dedicated to a really sweet friend that I met on that first trip to Uganda. She's actually a Kenyan-born journalist, Elizabeth Marab, world recognized for her scientific and health writing. Unfortunately, she succumbed to sickle cell disease complications just over a year ago in 2023. And it was during one of our first conversations, one of the first talks I got to hear Elizabeth give, that she had said, you know, I just got asked to write this article on stem cell gene therapy to cure sickle cell disease in the US and Europe. And the only thing more disheartening than being told you have a disease that can't be treated is to find out that you have a disease for which there are cures available, but just not available to you. And in honor of Elizabeth, fellow Ugandan patient advocate Evelyn Harlow and I published a viewpoint. This was an incredibly long, year-and-a-half-long peer-reviewed piece in Nature just last July on why it is so important for us to consider manufacturing these gene therapies and components in lower income nations in order to really make them accessible. And this is important because it's actually written into the International Covenant on Economic, Social, and Cultural Rights, Article 15.1b, that was signed in 1977, but never ratified by the US. This was implemented after World War II, and the acronym that they used was REBSPA, which is that everyone has a right to enjoy the benefits of scientific progress and its applications, and we're actually failing in our humanity if we decline to do so. And I think in the context of the upcoming administration, it's really important for all of us to remember that this is still a moral obligation that we have to the science that we're producing. Lots and lots of people helped with this work. I'm highlighting just 3 research technicians in the lab who I didn't specifically call out during the talk: Katrina Polyakov, Shirley Dwan, Molly Cassidy, who were involved in every one of the projects that you saw today. I also didn't have time to talk about a collaboration with Dr. Daniel Blanco-Mello in the Vaccine and Infectious Diseases Division at Fred Hutch, where we are actually looking at those intrinsic and extrinsic factors in HSPCs that are stymieing genetic engineering and evolving new ways to modulate them, in particular inside the body, as well as a collaboration with Dr. Umit Gurkin, who's a bioengineer at Case Western Reserve, to develop technology that can very, very rapidly sort stem cells from whole blood so that we can do it in a very short time frame, less than 2 hours, to envision something like an ex vivo process with in vivo-like timing. In other words, a single office visit. With that, I thank you for your attention. Questions?
I mean, in the grand scheme of things, this is a very, very trivial question. I'm just wondering if there's any chance the cells can exclude like double or triple transductions from the hybrid virus you made. Because when you said if you gave MOI of 10, you get 10% transduction, you give MOI of 20, you get 20% transduction, and give MOI of 50, you get 50% transduction. But mathematically, that shouldn't happen.
You're right, but it does. And I can tell you, we've just started looking at colony-forming cells and understanding exactly how many integrations per cell we're seeing. And we have a really high proportion of single integrants per cell. So something other people have shown, and we've seen this in the lentiviral space for a really long period of time, is that you have sort of this curve, right? You have a lot of cells that don't get any transduction, and then of the cells that do, a lot of them have one, some have two, maybe if you have three. I think we go up to like 5, and then we have this weird jump where then you get a few cells with like 8, 10, 12, and some ridiculously high number. And that's pretty much held true for both gamma retroviruses and lentiviruses in this space once you dial in on the optimal transduction level. We're just in the beginning phase of assessing this, but what we're seeing is that a majority of the cells have 1 or 2. We haven't found any yet with the high level of integrants per cell. It doesn't mean that they're not there, we just haven't seen them yet. I have spent a lot of time talking with Boro about how mathematically impossible this should be, and whether or not we need to revise the models or better understand what the heck this hybrid envelope is doing that's facilitating transduction so mightily for this particular F4 vector. I don't know the answer yet.
You don't know where the virus goes in the cell, right? With these 3 different.
We have some ideas.
That's probably part of the story, right?
Yeah, I mean, we definitely see punctate staining that is indicative of this being going in through an endosomal pathway. It's still going in through an endosomal pathway. That's the earliest data we have. We may just not see enough fluorescence intensity with the confocal to see smaller, individual particles crossing a cellular membrane or being more diffusely entered, because it's a really low MOI. We'd probably have to really hit the cells with a high viral load in order to really dissect that, but then we don't know if we're changing the mechanism of entry by doing that.
Who is next?
Great talk. I'm wondering if you've combined the hybrid envelope with any kind of enhancers or proloxamers to use a lower level of LV, but then still get maybe a little bit more virus into the cells?
We haven't yet. Even at an MOI of 50, we're in a pretty good spot for a single hit for the amount of transduction that we're getting. We're also really happy with these early results of 1, maybe 2 integrants per cell. So the issue with transduction enhancers is it's always a finely tuned balance, right? You might get a few more cells transduced, but if then you push a few cells into that 8 to 12 integrant range, you're just increasing your risk of genotoxicity. Now there's some, for diseases where you're gonna need a high number of cells transduced to see therapeutic value, where that may be worth the risk to benefit ratio. But there are a lot of applications where it's not. And we're really trying to stick with this idea of keeping it as simple as possible, as minimal as possible. Short, sweet, as few materials and reagents needed as possible. Caring Cross has their own mission. They've spun out a B corporation called Vector Biomed, which is trying to provide vector at a much more affordable cost. They've really revolutionized the lentiviral engineering and production pipeline. We're getting these vectors at high 10 to the 9th, low 10 to the 10th titers. That's massive improvement in production. That will buy us a lot to begin with. And so we're sort of okay sitting in the MOI of 50 range. Other trainees, I just want to make sure.
He's still training. Believe me, I'm training him still.
You all heard that. Especially David just announced he's in charge of Bruce. So if you have issues with Bruce, talk to his mentor, his training mentor.
That's right.
David.
Absolutely. He'll ignore it. So a couple quick questions. Beautiful talk. I enjoyed it. With your measles cocal virus, it's going through an endosomal pathway. Mm-hmm. Measles tends to be a non-pH dependent pathway.
Right.
Comments?
I don't know what to tell you. We honestly don't know how this is doing so well. It's not what we expected. We totally expected to see transduction at 24 hours, given that it was an endosomal pathway, do better. It's maybe that the endosomes that include LDLR are different than the intracellular vesicles that are taking up CD46,
Could be.
although it should be an endosomal
We
pathway.
found different pathways with them. We can talk about this offline.
Yeah, totally. And I'll say, we didn't have time to talk about it, but we've looked at a lot of the stem cell markers, CD90, CD133, CD117, and they also use different endosomal pathways when they internalize after engagement. CD90 in particular uses like a microvesicle that traffics directly to the endoplasmic reticulum versus a pH-dependent endosome. We haven't done a lot to look at what kind of endosomes. Are these clathrin dependent, which is most likely what is happening when we see this, but we don't know that yet.
So, we found that basically with VSVG, you have to get activation to get recycling to occur.
Right.
Do you see recycling when you bind, or is recycling occurring on your freshly thawed cells?
Yeah, so we looked at this question with the cocal a little bit because we just saw such a different transduction efficiency with the cocal than VSVG, and they should be using the same receptor.
Right.
And the answer was we do see it, but it's different, right? So right after thaw, we see the cells kind of like waking up. The membrane's not totally intact, right? These have been cryopreserved cells. But we also see that they right away kind of like, where am I? What am I doing? Let me take in some signals and see where the heck I am and what's happening. And so we do think that there's a little bit of probably micropinocytosis facilitating some of this uptake, which will not be a pH-dependent mechanism. It is, in some ways, we're just super lucky. It's like this thought, try this experiment, and then somehow we're getting really, really efficient, quantitatively predictable delivery with this hybrid that we didn't expect to see.
One last question. In the Magenta's work, they were looking at insights, if I remember correctly, to knock the cells out of their space to allow repopulation.
Mm-hmm.
Do you see a similar phenomenon with some of the stuff you're doing, exiting out of that stem cell space where the stem cells can then sit back in?
Yes. So every one of those mobilization agents that we're testing, we've shown in a peak mobilization study has the capacity to do that. So they're vacating the niche for a short period of time and then returning to the niche. So refilling that niche.
Thanks so much. Great
Welcome.
talk.
Yeah, I'm really excited on your excellent results. It's wonderful. So my question is that in your intraosseous delivery of the gold nanoparticles, so I might have missed it, but are they targeted towards hematopoietic stem cells? Are you? But you do see quite a good because with lipid nanoparticles, sometimes they have to put, targeting moieties.
Yeah.
So do you think if you add targeting moieties, you may enhance your editing?
Yeah, I just for sake of time didn't have time to show the data. We analyzed all those different receptors on stem cells that I talked about because we're looking for those that induce a receptor-mediated endocytosis that we could hijack essentially to carry the particle into the cell. And what we have found is that there are at least 3, 133, 117, 90. Those are all reported to endocytosis. No surprises there. We were surprised to see that CD90 appears to be using a different mechanism of cellular entry. But there are also there's another issue, which is that those markers are not unique to stem cells in the body. So when we do it in a humanized mouse, it's an artificial system because the only cells expressing that are the human cells that are in that mouse. But when we take this into a human, there's going to be a lot of cells expressing CD90. It's on every T cell. It's on endothelial cell progenitors. It's going to be actually lining vascular circulation. So that may not be the best marker to use, even though it looks like in this defined model it could be better. Same for CD133 and 117. These are not just hematopoietic stem cell markers, they're stem cell markers, which means they're on stem cells all over the body, including in the central nervous system and in particular in the gonads. And one of the bars here is thinking about how this is going to impact fertility and future generations. When we talk to sickle cell patients in particular in these low resource settings, they are super excited that we have a cure here in the US, but the second they find out that it's basically a bone marrow transplant, they're no longer interested because fertility is a huge part of the social paradigm. And if they cannot maintain the fertility of their child after they go through this therapy, they're not interested. And we are going to have to address that. It's a social issue, but we have to address it in making these therapies reasonably accessible. I highly encourage everyone working on a therapy or thinking about this, please talk to the people who are the end users of this approach. One of the most poignant vignettes I heard from someone else was around a gene therapy for muscular dystrophy that had been developed for over 2 decades by a group in California that had gotten a considerable number of resources, fine-tuned this AAV delivery system to hit cardiac muscle in the diaphragm because these patients die of cardiac or pulmonary failure. And they thought, great, they got the clinical trial through, they did an IND, they went through the regulatory body, they brought it to the patient groups, and the patient groups said, no, thank you. And when they said, why? The patient said, I'm already at the place where if I die tomorrow because of heart failure or pulmonary failure, that's okay, because I don't want to live another 20 years with a colostomy bag. Can you please design a gene therapy that gives me back bathroom continence? And so if you don't know what the patients want, you may be spending a lot of time doing something really clever and cool that nobody's going to take or that is not going to be reasonably translatable to greater society. And I'll just throw this out there. Solving the problems to actually make these therapies work for the most difficult populations is going to solve the issues with cost and sustainability that we have here in the US. There's a great economist named Christian Claytonson. He's probably most well known for the innovator's dilemma. He was at Harvard Business School for a while. He actually just passed away in 2020. Highly recommend a great talk where he went to Google, and they have it on YouTube. You can watch it for free. But another book that he wrote was called The Prosperity Paradox, How Innovation Can Bring Low Resource Settings Out of Poverty. And he really emphasizes this point that the greatest innovation comes from the places that have absolute need to solve these problems, more so than in spaces where we think about it as a luxury.
I think we're over time, but I was going to end with a question related to that, which is
Go for it.
as you reduce the cost of goods and the efficiency, in this process, and we get therapeutics out to more patients, there's also this issue of rare disease where the companies are all leaving the space. And so how do you, I think it's sort of related to your last comment, but how do you bring this back around to, we all take care of rare diseases. Most of our pediatric diseases are rare, but the companies don't want to invest in them. How do we take advantage of this and move it towards rare disease?
Well, I would say, the paradigm that rare disease spaces for which there are no alternatives, and we do need to make these therapies accessible. The withdrawal, we call this now gene therapies have shown us 2 valleys of death. There's the development valley of death where we say, okay, a technology just couldn't get the investment it needed to get to a clinical trial, and that's called dying on the vine. Now we have an economic valley of death. It got through commercial authorization, it got through all the trials needed, and it got through regulatory approval, but now it's not making the company enough money in a fast enough and more predictable enough time, and so the company just drops the pipeline even though all the safety and efficacy data is there. This is also a complex problem that is driven by healthcare systems that are capitalist. And it's not a super easy issue to address. It comes from my least favorite economist, Milton Friedman, who really popularized this idea that the sole purpose of companies is to increase value for their shareholders. This was an idea that if I'm a child of the Human Genome Project, I'm also a child of Milton Friedman economics. I'm from Ohio originally. My parents were a teacher and a steel worker. I am from the heart of the Rust Belt, where the coal, automotive, and steel industries declined because of Milton Friedman economic popularity. Companies, my grandfather, I would say, had a pension and a nice house and got a bonus when he had kids because he was giving more workers to the factory later on. My dad, who worked for the exact same steel company, who even had a higher level job, did not get any of those things. And the unions were dissolved because companies just decided, reduce the bottom line, increase the value of the product so that the shareholders make all the money, but only 50% of the country invests in the stock market. There's a lot of complexity to this. There are a number of models to address the rare disease problem. One of those is shifting our thinking from, if this is a transformative technology that has utility and cuts its teeth in a rare disease, but that is never going to be a commercially viable population for a company, can we create a new regulatory pathway for the FDA that allows a hospital to continue administering those essentially at cost, gets reimbursed from the healthcare companies for essentially administering those therapies so that they're accessible, but without a profit, so that they can continue to be developed. And then if an institution can create a bundle around a few more of those therapies for a bunch of rare diseases, when you have, the margin is sometimes 5 if there's enough rare with ultra rare. For entirely ultra rare portfolios, it would be closer to 10. If you have, let's say, a lentiviral vector where all you have to do is change the therapeutic gene, but it's the same vector and you can treat 10 different ultra rare diseases, that is a commercializable platform. And then you can out-license to a commercial entity. But you have to have the regulatory approval to be able to move that forward and to be able to give it to patients and have the hospital get reimbursed for that until you reach that bundle size. So that's one strategy, and that is being employed in the European system right now. In Spain, they are administering a CD19 CAR T-cell product that way through hospitals there, and now Italy is also trying to move some of its rare disease gene therapy forward with that model. 27 countries with one regulatory agency, European Medicines Agency. We are one country with one regulatory agency, and we don't know who's going to lead it for the next 4 years yet. So we have to think about the ways that we can kind of strategize these arguments. As much as I talked about economic policy, there is not really distribution widely to a large number of people who need it without a commercialization structure. Industry still is a necessary component of this, so we have to figure out how to work together more effectively.
Okay, thanks so much, Jen. Super job.